Imagine editing the very blueprint of life, not just a single gene but removing an entire chromosome. Recently, scientists achieved this in the lab, deleting the extra chromosome that causes Down syndrome. This is a massive leap in genetic engineering once thought impossible. The breakthrough using a gene editing tool to discard the unwanted chromosome without chaos. Instead of just treating symptoms, we might one day fix the root cause at the cellular level. In the experiment, once the extra chromosome was gone, the cells behaved more like typical cells. They grew better and functioned more normally, giving us a new way to study and potentially treat genetic conditions. This is just the beginning. These are lab experiments, not treatments for people yet. But every revolution starts with a single discovery. This proof of concept challenges old limitations and opens new possibilities. It's not just about one condition, it's about mastering our own biology. And now we know it's possible to delete an entire chromosome. To see why this matters, let's look at Down syndrome. Most people have 23 pairs of chromosomes, 46 in total. Down syndrome happens when there's an extra copy of chromosome 21, trisomy 21. That extra chromosome disrupts development, leading to the traits and health challenges of Down syndrome. Hundreds of genes are overexpressed, throwing off the body's balance. This causes developmental delays, intellectual disabilities, and higher risks for heart and immune problems. For decades, care focused on managing symptoms, not the genetic cause. The idea of removing the extra chromosome was just theory, until now. This new research tackles the problem at its source. It's a completely new way of thinking about genetic conditions. And it all starts with understanding the impact of that extra chromosome. How did scientists do it? With CRISPR, a revolutionary gene editing tool. CRISPR acts like programmable molecular scissors, able to find and cut specific DNA. It uses a guide RNA to target a DNA sequence, and an enzyme called Cas9 to make the cut. Before CRISPR, gene editing was slow and expensive. Now it's fast and precise. Scientists use CRISPR to study diseases and develop new treatments. But this experiment went further. Instead of editing a single gene, they aimed to remove an entire chromosome. By making a large cut in a key region, they destabilized the chromosome. The cell's own machinery then ejected the damaged chromosome during division. This was a bold, demolition-style use of CRISPR. It shows how far gene editing has come, from single genes to entire chromosomes. This approach could change how we tackle complex genetic disorders. It's a new era for genetic engineering, and it all started with a precise cut. Here's how the experiment worked. Scientists took skin cells from people with Down syndrome and turned them into stem cells. These stem cells can become any cell type, making them perfect for testing. Using CRISPR, they targeted a region on chromosome 21 for a large deletion. The damaged chromosome was flagged and lost during cell division. After treatment, many cells had only 2 copies of chromosome 21, trisomy corrected. Genetic analysis confirmed the normal chromosome count. Most importantly, the corrected cells grew and functioned more like typical cells. This proved that removing the extra chromosome could reverse cellular effects of Down syndrome. The experiment moved the idea from theory to reality. It's a landmark achievement in genetic research, and it opens the door to studying and potentially correcting other chromosomal disorders. This breakthrough is a game-changer for research in medicine. Scientists can now compare corrected and uncorrected cells from the same person, isolating the effects of the extra chromosome. This helps us understand how trisomy 21 affects different organs, like the brain and heart. It could lead to better targeted therapies, even without direct gene editing. Until now, gene therapy focused on single gene disorders. Tackling a whole chromosome was out of reach. Until this study. It shows our tools are powerful enough for complex genetic errors. This could inspire new strategies for other chromosomal conditions. The ultimate hope improving cell health to support better lives for people with Down syndrome. It's a new platform for discovery and optimism. This is early-stage science, done in lab dishes, not people. Turning this into a therapy will take years, with huge technical and ethical challenges. Delivering the treatment safely to trillions of cells is a massive hurdle. Down syndrome is part of people's identities. The goal is to improve health, not erase diversity. Any future therapy must be guided by the Down syndrome community. For now, the focus is on refining the technique and learning more about trisomy 21. This research is a tool for understanding, not a cure yet. As gene editing advances, we must ask, what should we change and why? The promise is huge, but so is our responsibility. We're at the frontier of medicine. How will we shape the future?